So, sorry, be a bit of a change in style. First of all, I'll be using the dreaded PowerPoint, but I promise to go back to the blackboard in the next lecture. What I'll do actually is give you a bit of the biological background and motivation and context for why I was given 5 lectures on evolution of sex and recombination. In fact, I won't be just doing that. I'll be today in this lecture explaining why we're interested in that question and why we're interested more generally in the evolution of genetic systems. And then tomorrow, and for a bit of the next lecture, I'll be talking about the population genetics of multiple loci. First of all, actually taking a detour from this topic to talk about what happens when the gene pool is well shuffled, when we have a so-called beanbag model, which is the relatively simple case, and then moving on to ask about what happens when we have correlations between loci, that is linkage disequilibrium, and talking then specifically about how we can explain, using population genetic models, evolution of sex and recombination. So today is really setting the biological context motivation, and then I'll be doing the population genetic theory next time, and then moving on to actually explaining sex, life, everything. Okay, or not, depending. See how far we get. Okay, so background reading. There is an excellent textbook on evolution, which I wrote with other people. So there's no advertisement here. And it has the imaginative title of Evolution. I couldn't think of anything better. But chapter 23 specifically is about evolution of genetic systems, including evolution of sex. And that is on its way to the web at this moment, I think. It's on a memory stick at least, so it will appear. And there's a couple of other chapters from the textbook on random drift, on interaction between different evolutionary forces, which might be useful as background. But chapter 23 is really the main one I'm putting up. And also the more general stuff I'll talk about today, a lot of this comes from a really great book called The Major Transitions in Evolution, by John Maynard Smith and Urs Schaffmari, which I can't pronounce properly. So this book was 1995, I think. And actually, if I remember, I'll put up an article. They managed to condense the whole book down to one article, which they published in Nature at about the same time. And this covers a whole range of big questions in biology, but including a lot of what I'll talk about today. So, basic question is, why are organisms the way they are? And maybe before I put up a list of questions here that kind of came to my mind, maybe what comes to your mind? What would you say would be an interesting question, a puzzle? A big question about why are organisms the way they are? What do I mean by that? What do you think I might mean by that? What sort of question comes to mind? Okay, yeah, that's a great question. So we could say why species, right? Why do organisms cluster into particular sort of types that you can give names to? Okay, that's really a population level question about the relation between many individuals. There are also questions about individual organisms and questions about their genomes and the way the genomes work. Other questions? Yeah. Yep. Yeah. Why do the cells bother getting together and cooperating to produce, well, us? Sorry? What? Party? You think it's just fun getting together? Are they having fun, our cells? And you could include aging in there, right? So, yeah. I actually brought a book with me. It's light reading on the airplane called Sex and Death, but it was written by a philosopher of biology, so it's actually rather dry. But obviously, we're really hoping to counterbalance that with a good title. Any others? It's interesting. So these are all, really interesting, obviously very deep questions, and they're almost completely disjunct from the questions I had, which are probably much less interesting. But let's see. So I was thinking about things specifically about genetics. Let's narrow it down, and why are genomes so big, right? Why do we have 3 × 10⁹ base pairs rather than some other number? Why do we have 22,000 genes rather than 50, 60,000 like an advanced organism like maize, corn? We actually don't have any a priori argument, I think, for why genomes should be the size they are, or the number of genes should be the number they are, etc., etc. While in eukaryotes, we have this bizarre system where the coding sequence in the DNA is broken up into exons that do coding and then introns in between, which just sort of make life more complicated. And then you have to go through a whole process of transcribing a long RNA sequence, which is then cut and spliced to produce the more compact coding sequence in the messenger RNA, which then is translated. It just seems, you wouldn't design that from scratch. So how did that arise? Is it adaptive? Is it just kind of arbitrary? What's going on there? Why 2 sexes? Right. So this is an arbitrary list. I mean, it's probably just as arbitrary as the list on the blackboard. Why 2 sexes and 3 sexes? There's actually a famous quote by R.A. Fisher, which is, if you really want to understand why there are 2 sexes, typically in familiar organisms, you have to think about the consequences and the evolution of bizarre possibilities, like having 3 sexes or 4 sexes. And so another question, which actually Urshad Murray wrote a really interesting set of papers on, why are there 4 letters in the genetic alphabet? In other words, you read along the DNA and you have A, T, G, or C, 4 all possible bases, and that codes for the protein sequence and also determines binding of this and that to regulate gene expression. Why 4? You could have a system where you just had A or T, 2 letters. Okay? And there'll be information, there'll be half as much, but in some ways it would be simpler. You could have 6 letters, and in fact there have been experiments producing modified bases which can actually be replicated. You can produce a 6 or 8 letter alphabet in principle. Okay, so why 4? Even deeper question, why should heredity be digital, right, and linear, a sequence of 4 possible states? We're used to that idea, but maybe if you put your minds to it, you could imagine a 2-dimensional sheet of digital states. Not quite sure how the replication would work. Or you could imagine some kind of continuous variable rather than discrete states. Would that work? Could we be analog instead of digital? Yeah. Well, is it? I don't know. I mean, Schrodinger's equation is a continuous space. So, I mean, then you get down to particles and really, really deep down. But you could imagine, all right, you could imagine a system with, if you like, many, many letters, and therefore almost a continuous range of possible states, which would be sort of moving towards continuous. Yeah. I mean, the way people thought of blending inheritances was a continuous process. It was like mixing paint. So, these are just questions that come to mind about, why are things this way? And some of them could fall into the class of evolution of evolvability. Probably not these, but certainly why sexual reproduction and multicellularity, possibly. Questions actually I didn't raise, and we've actually heard already, people talk a lot nowadays about modularity to the extent that, not all genes are expressed at all times. They're expressed in specific times and places in a multicellular organism or even in a unicellular organism, and seem to be organized in pathways that are somewhat modular. There's a pathway for doing this, there's a pathway for doing that. I'm never quite sure how modular organisms really are, because of course there's a bias. Insofar as we can understand anything, it is rather simple and has a rather small number of connections. So there's a little bit of a bias towards thinking things are modular, because that's the only way we can understand them. But to the extent that organisms are modular, why should that be? Another term we've already heard, I think yesterday, were terms like robustness, canalization, redundancy, all these features of organisms which mean, for example, in the eukaryote, most of the time you can knock a gene out and you have no phenotypic effect. So it's robust in that sense. Why did that evolve? Is it just kind of the way the organism was constructed, or was it selection for that particular degree of robustness to both environmental perturbations and to the effects of random mutations. So these sorts of questions are very closely, well, they can be organized under the heading of evolution of evolvability. And one way to think about this is to think about the analogy with computer science and computation. And there's a big field in computer science which started with John Holland in the 1970s, who saw an analogy with the power of natural selection and the amazing apparent design of living organisms, and thought, why don't we apply this to producing algorithms or solving complicated problems in general? And the basic idea is very simple. You've given up on coding your particular algorithm for doing some task, and instead you just produce a set of algorithms that work more or less, and then you mutate them, or you recombine them. You take two of them and you sort of shuffle the code, and you produce new ones, and then you select the ones that work best. And you keep doing this, and this actually works. And there's a huge sort of field with several conferences each year of so-called evolutionary computation, where people just use natural selection, essentially, I should say artificial selection, to develop better solutions to problems, better algorithms, better programs. And it's quite interesting that although it was started by analogy with biology, it's gone off in its own direction. They almost never communicate with biologists. There's a little bit of communication starting up, but they've developed their own theory, which looks completely different from our population genetic theory. It's kind of independent replicate of population genetics, and it's kind of a little bit disturbing that it looks quite different. It's true, but it's just asks different questions. But the reason I'm bringing up this whole evolutionary computation business is that actually, if you take a standard programming language, Fortran or C or whatever, and you take a program and another program, and you recombine them, or you randomly mutate them, mostly they just crash. Right? They're not evolvable. And so the main problem in this evolutionary computation business isn't actually choosing the selection strengths and the population size. They do mostly the same things we would do in a population genetic simulation. The real problem is representing the problem in the right way, such that you can mutate and recombine and still get something that works. And that's what we might call the evolvability. You have to come up with a way of building their computational organism, okay, or building a biological organism, which is evolvable, in which you can make changes such that the thing still functions and has an appreciable chance of functioning better. And the big question is, to what extent is this evolvability simply a side effect of having an organism that functions at all? And to what extent is it something that has itself evolved as an adaptive functional form? And these are quite subtle and difficult questions, and I think there are big differences in mindset between, on the one hand, evolutionary biologists, and on the other hand, molecular biologists, in how far they assume or take for granted that something is functional and has evolved for that function. And I'll try and explain some background, some historical background about why these issues are actually quite tricky, and why actually evolutionary biologists are really quite cautious about saying that something is an adaptation, and in particular an adaptation for evolvability. And so I'll be focusing in the future lectures on evolution of sex and recombination, because that actually is the best understood and the longest studied example of an adaptation for evolvability. Because it seems pretty clear that sex and recombination have actually evolved in order to facilitate the evolutionary process. But that's something that's quite hard to establish, and there's been a lot of argument about it, as we'll see. So any questions at this stage? This sort of general, I'll get more specific. Okay, so if we think about problems involving evolution of evolvability, something I hadn't mentioned yet was maybe the simplest case to understand is evolution of mutation rate. So, of course, mutation is inevitable. You can't have a perfect system for copying the DNA sequence from one generation to the next. There will be some mutation. But the question is whether the mutation rate has evolved in order to facilitate the production of beneficial mutations, because, of course, if there were no mutations, there would be no evolutionary change, there would be no adaptation. Or, on the other hand, whether actually most of the time mutations are bad, and mutation rate is simply set by a trade-off between trying to selection to reduce the mutation rate and the costs, the metabolic costs and so on, of having a more and more elaborate repair system. Okay. And that's quite unresolved. And even in that simplest case, it's really not obvious to what extent the mutation rate is determined by simply selection to minimize copying errors, or possibly by selection for evolvability in some sense. Sex and recombination we'll be dealing with in detail, and I've mentioned other things like modularity, like robustness. Additivity I put in there, and that's something that will hopefully you'll understand a bit better after Bruce's lectures on quantitative genetics. There the argument is that alleles can be picked up by natural selection in a sexual population only if they have a consistent effect. In other words, if you have an allele which is good in this genetic background and bad in this genetic background, then essentially natural selection won't be able to get a grip on it, because something that was good one time, at a later time as the genetic background changes, all the other genes in the organism changing at the same time, its advantage may disappear. And so, in a sense, selection is most efficient when a particular allele always increases fitness by some definite amount. Okay, and one can quantify that. And the question is, why is it that actually most of the variation in populations that we see can be attributed to these consistent additive effects rather than conditional epistatic effects? And this is actually a puzzle. Okay? People argue about this. Okay, so how do we go about, in general, understanding why organisms are the way they are? It's simple if you have some sort of very straightforward problem where there's a clear optimal way to be, like, let's say, the vision of an owl, which is extremely efficient. It's close to the physical limits of what can actually be achieved optically with something like vertebrate eye. And we can understand these sort of engineering principles. We can understand how selection is favoring this particular structure. And you make the same kind of arguments about bird flight, about the way the feathers have to be shaped, the way the musculature has to work, and so on, to allow the bird to fly efficiently, using sort of aerodynamic and engineering principles. Although actually even there we get into kind of difficulties, because a lot of bird plumage is about looking good to the other sex. So there's kind of sexual selection coming in, which is often conflicting with simple optimization. So actually optimization principles only take us so far in biology. But actually historically, there's been, it's much harder actually to understand these questions I've been putting up, the bigger questions about why organisms are the way they are in some broader sense, and in particular to understand features of organisms that are favoring future evolvability. Because at first sight, it seems really hard to see how that's an advantage to the individual. And historically, people appreciated way back, in fact, the first person to clearly write about these issues, I think, was August Weismann in the late 1890s, around early 1900s. And he articulated very clearly that sexual reproduction was a good thing in the long run because it generated new variation by shuffling attributes of different organisms. And I think Darwin appreciated this as well. So in a general sense, people appreciated that somehow sexual reproduction generated novel combinations of characters and was good for the species. And this was kind of accepted without people thinking about it very clearly. And in fact, many, many features of organisms were just accepted as being the way they were because it was just a good thing in the long run for the species. And this changed really in the 1970s, stimulated partly by a book by Wynne Edwards, who was really an ecologist, who argued explicitly that species of birds, say, would evolve to reduce their reproductive capacity so that they wouldn't overexploit the environment, okay? Which would be a good thing for them to do in the long run, but it doesn't make any sense for an individual to produce fewer offspring because of some future benefit or some future consequence. And so a lot of evolutionists, particularly George Williams and John Maynard Smith and Bill Hamilton to some extent, sort of really saw that there was this complete misconception about the way evolution worked. And they, in a way, went back to a much more kind of pure Darwinian way of thinking in which you had to justify evolutionary processes in terms of differential reproduction between individuals. And actually show that the mechanistic processes of selection, mutation, recombination, whatever, would lead to the outcome we see. Not simply saying, well, it would be good if we had this, it would be good if we had that. So, sorry, was there a question?
So the thing is, what does it mean to say, like, you're basically saying a particular species evolves let's say someone argues that a particular thing has come about, so the species it is beneficial for the species. The point is, let's say I basically call a particular species based on their genome. If that is changing, then the species itself is changing. So the thing is, what does it even mean to say that it's for the benefit of the species when the change has made the species? I mean, like, it's a different species now.
So you're actually bringing together two very confusing and complicated arguments. And the second one is, what is a species? And can we say what's good for the species when the species itself is changing and may split into other populations and so on? Yeah, indeed, that's a good point. And it's actually one of the difficulties, I think, in making a sort of good of the species view coherent, because the species isn't a fixed thing. What you have is collections of individuals. So in a way, that's a good point. It's one of many criticisms of this for the good of the species thinking. So people in the 70s started to think about these things more clearly, and actually realized also that you couldn't just go around saying that this or that was an adaptation. And they started to define much more precisely what's meant by an adaptation. And I suppose I'm not going to give a very clear definition which will get past the philosophers. There's a whole field of philosophy of biology which deals with these kinds of issues. But the point is that you have to not only show that some particular feature or aspect has a function, okay, which increases survival and reproduction, but you also have to show that it evolved because of that increase in survival and reproduction which it produces. And of course, many features actually first evolved for one function and have been co-opted for a different function. So it's not at all obvious exactly what the selection pressures were that produced some particular feature that we see now. So let's take an arbitrary example about which we actually don't know anything very much, probably not a very good example to take, but the evolution of feathers, which help birds to fly. They may have originally evolved for display or for thermoregulation or whatever, right? It's clear that the first feathers weren't there to help birds fly, because there's a whole chicken and egg problem. And when you start thinking about major novelties such as the evolution of flight, if you try and think, well, how did all this come together for this purpose? It's impossible to imagine. Indeed, there was presumably some kind of complicated series of events in which something evolved for one purpose, was co-opted for another purpose, was co-opted for another purpose. Very obscure chain often, but we can in some cases piece it together quite well. And at the end, you have something which has quite complicated origins. And so generally through this time, through this, a lot of argument about what people saw as a sort of earlier, more naive attitude to explaining adaptation, evolutionary biology generally became very cautious about attributing adaptation to particular functions. And that persists as a kind of attribute of the field generally. So it became clear at this time that we had to have theories that were based on population genetics, that were based on ultimately selection amongst individuals, okay? And a particular approach which turned out to be very important for thinking about evolution of evolvability particularly was to think in terms of modifier alleles. And this is a kind of a thought experiment. It's not that anyone thinks these modifier alleles necessarily really exist, but you're thinking, suppose a new variant came into a population, a particular allele, a particular gene, which, let's say, increased the rate of sexual reproduction or increased the mutation rate or changed some other feature. So a modifier is, in its purest form, has no effect except to change the particular feature you're interested in. And you ask, would that allele increase in the population or would it decrease? And so this is trying to pare down the argument to the simplest possible scenario, asking, would alleles that increase recombination, that increase sex, that increase mutation rate, or whatever, would they increase in the population? And ultimately, you have to have some kind of argument like that in order to explain the origins of these kinds of features. And as I go through in the later lectures, I'll be giving specific examples of how we use this idea, but just introducing it as a way of thinking about how we explain features that appear to have only a long-term benefit, but can actually be explained in terms of short-term changes. So that's kind of a roundabout explanation of why I'm going to focus on sex and recombination. So it's great, the timer is still on 1 hour, 30 minutes. I can go on forever. Sorry? All right, I'm disappointed now. Okay. So So what is sex, right? And again, I'm not going to try and give a very clear definition of it, but we could say the mixing of hereditary material to produce new genotypes. Actually, it's not so obvious what I mean there, because you normally think of sex as being between two different individuals, right? A mixing of their different genetic material to produce offspring which are distinct, and with eukaryotic sexual reproduction with crossing over across the whole genome. Actually, almost always an individual that's never been seen before, a new combination of the alleles in the two parents. That's what we normally think of. But actually, you can think of sex within an individual. What I mean there? Well, self-fertilization in a hermaphrodite. So I should say the physicist should stop me if I use technical terms. There's a lot of technical terms here, which are really rather unnecessary, but a hermaphrodite is just an individual that combines male and female function. If you think of flowering plants, then flowering plants typically have flowers that contain both the male organ, the anther that produces pollen, and the female organ, the stigma that receives pollen. Pollen tube grows down and fertilizes the ovule. Okay? There's actually a whole variety of mating systems in plants, which are really fascinating. Some plants have a hermaphrodite individuals, but they have the separate male and female flowers. Other plants are actually what we call dioecious. They have male plants with male flowers, female plants with female flowers, and so on. But most flowering plants are hermaphrodite. And some fraction of those can self-fertilize. Simply take pollen from the anther within the flower, and that pollen simply falls onto the stigma and produces a new offspring. And that offspring will be different from the parent, because let's think about just a single genetic locus. Let's suppose that the parent was heterozygous, big A, little a. Then if you have self-fertilization, half of the pollen grains will have big A, half will have little a, and half of the ovule will have big A, half will have little a. And so we have the famous 1:2 ratio, that the offspring will be a quarter big A, a half big A, little a, and a quarter little a. So you've produced new genotypes, and if you think about many loci, you produce a whole lot of new genotypes, you produce variation. But we're kind of not very interested in self-fertilization in the long run, because you can see that after one generation of selfing, you'll have half as many heterozygotes. After 2 generations, you'll have a quarter as many heterozygotes, and pretty quickly, you'll end up with a single genotype, which is completely homozygous, and then there will still be sex. There will still be the male gamete, well, still be meiosis, producing male gametes, meiosis, producing female gametes. Those come together within the flower to produce a new offspring. But because the two parent genomes are now genetically identical after many generations of selfing, there's no actual genetic consequence. So really what we'll be talking about is outcrossing sex, okay? Where 2 genomes from, or genetic material more generally from different individuals comes together to produce new offspring. Now I've drawn here the sort of typical and the most familiar kind of life cycle, kind of sexual life cycle, which I'll be concentrating on. And it's important to realize that it involves an alternation necessarily between a haploid and a diploid phase. And this is sort of necessarily true if we're going to have sex in which 2 gametes, okay, carrying a single copy of each chromosome. So this illustrates 2 chromosomes, each present in one copy, a black and a gray one there. They come together, they produce a diploid. So this process is called syngamy, that's fertilization, that's coming together of pollen or ovule or sperm and egg. It produces a diploid individual, and at some stage that diploid then goes through some kind of meiosis, which produces, again, 2 haploid, or a number of haploid gametes. Okay? And this process of meiosis may or may not involve crossing over to produce recombinant chromosomes with a mixture of material from this parent and this parent. So we have an alternation between haploid, diploid, haploid, diploid. And we sort of think of ourselves as human beings as proudly diploid, right? But actually, through our life cycles, we all have, the males will produce sperm, which are haploid, and females will produce eggs, which are haploid, and so on. So there's a very, very brief period of haploidy in the human life cycle. In plants, there's a slightly longer period of haploidy because the pollen tube in its development, sorry, the pollen grain in its development goes through a number of divisions. Actually, sperm also go through a number of divisions. There's a very short phase of actually haploid reproduction, asexual somatic reproduction. But actually, there are many organisms which go through most of their life as haploids. Many fungi, for example, will be mostly haploid and go through a brief sexual phase involving diploidy. Others will have more or less similar stages, which may look similar morphologically or may look quite different. Many algae have, alternating haploid-diploid phases and have very interesting life cycles, much more interesting than our rather dull, predominantly diploid. So there are all sorts of questions about, why is this, which I'm not going to really get into. But you look at this, and this is the standard sort of textbook, classical genetical life cycle, familiar in Drosophila and humans and whatever, in animals, in plants. Not necessarily, well, maybe something very vaguely like this is typical across eukaryotes. Sorry, is everyone, I'm using the word eukaryote, prokaryote, so on. Everyone okay with that, including physics people? A eukaryote is simply one of the three branches of life which has a complex cellular structure, including a nucleus, hence its name, but also including mitochondria, and in the case of photosynthetic organisms, chloroplasts, and generally a much more complex cellular structure than bacteria or archaea, the other two major branches. Okay, so how did this start? Well, actually, I'm going to try and be avoiding questions about how things started, because we don't really know, but there's some thoughts on it before moving on to what we can say much more about, which is how the sexual life cycle is actually maintained in the present-day world. But how can we imagine this thing started? Well, actually, there's a very straightforward and immediate advantage to syngamy, to two haploid cells coming together and producing a diploid. And this advantage is simply that if we have a population of haploid organisms, as I think Bruce said yesterday, they'll accumulate mutations. Most of those mutations will be deleterious, and they'll be weeded out by selection, but there'll be some frequency, which is roughly mutation rate divided by selection coefficient, a frequency at equilibrium in a balance between mutation and selection. And mutation rates per gene are very small. Selection coefficients are typically much stronger, otherwise the gene will simply degenerate and lose its function. So this frequency might be, let's say, if we said the mutation rate per gene of a few thousand base pairs was 10 to the minus 5 or 10 to the minus 6 at the most. Selection coefficients 10 to the minus 3. So we're now talking about, a 1% frequency of deleterious mutations. That's actually rather a high level. Mutation rates might be lower, selection coefficients higher, but still you've got a lot of genes there. And so typically, in a typical eukaryote, and this goes for all eukaryotes really, you'd expect there'd be, 10, 20,000 genes, something of that order, and the total number of deleterious mutations carried might be quite large. It might be, 100 or so. Obviously, it depends on the genome size and so on, but the cumulative number of deleterious mutations is quite large. Each of them has a small effect, so the organism survives, but, it's a bit of a mess. So, suppose two organisms unite and get together. They go through syngamy to produce a diploid. Well, there'll be twice as many bad mutations, but they'll be at different genes, and this is crucial. And typically, deleterious mutations are recessive, and there's actually a long argument about why deleterious mutations should tend to be recessive. But pretty much the accepted view now is that it's simply because most deleterious mutations are a loss of function, and a loss of function can be complemented by a functional copy in the genome. So if you have a diploid, losing one copy is not too bad. It's only when you lose two at the same site that you really suffer. Okay? And it is empirically a fact that most deleterious mutations are recessive. Just to mention, it's sort of relevant to this whole evolution of evolvability debate, that there's a huge argument between Fisher and Wright in the 1930s. Fisher thought that actually recessiveness evolved because it was kind of a good thing. He didn't have a good of species view. He had an individual-based mathematical model showing how actually there could be selection favoring recessiveness, basically because it would increase the fitness of the heterozygote. And Wright argued that, yes, it could work, but it was incredibly weak, the selective process. And they went back to the forwards. And one really key bit of evidence against Fisher's theory, in favor of the simple idea that it's just the fact that deleterious mutations are loss of function, is a nice survey, actually, by Alan Orr, who pointed out that if you look at Chlamydomonas, an single-celled alga, which spends most of its life as a haploid, you can make diploids in the lab artificially, and you can show that mutations in Chlamydomonas are indeed recessive, even though they've spent their evolutionary history predominantly in the haploid phase. Okay. And so you can't really come up with any adaptive evolution of recessiveness, given that a haploid organism has mutations that turn out to be recessive when made homozygous in a diploid. So anyway, that's a bit of a sideline. The point here is that haploid organisms with reasonable sized genomes will have a large number of deleterious mutations, weak effect, and they'll gain an immediate and very, very large advantage when you put two of them together. And actually, we see this. It's very important in crop breeding. So most of the corn that's grown is actually hybrid corn, because different, somewhat inbred strains of corn, when hybridized with each other, produce a diploid with deleterious mutations derived from the different populations. They gain a big advantage. Okay, so and this is a really big effect, very general effect, so-called heterosis. So this heterosis gives an advantage.
End of the day, like let's say there is a mutation. So what matters is at the end of the day is basically the protein they code for and what function it does. If the protein structure, so since protein function is strongly dependent on the structure, if whatever the mutation that occurs changes the structure significantly, then you can say that that mutation has an effect. So the point is, if there are protein structures which are robust to, let's say, even deleterious mutations, so the function is not so particular about the particular structure of the protein, in that case not be like sort of recessive or anything.
That's true, and there will be you will perhaps tend to evolve proteins that are robust to mutation. That might happen, and indeed most mutations to proteins don't destroy their function. That's the kind of basis of the neutral theory of Chimera. But it's still true that there are some mutations that destroy their function. So we're talking here only about deleterious mutations, and it's still true that deleterious mutations typically are recessive, because if you have a protein and it has a mutation in the small fraction of sites that are important, okay? So in the active site of a mutation, it destroys the enzymatic capacity. Then if you've still got a functional copy coded by another copy of the genome in a diploid, then you're okay, because you have enough enzyme function. Now, of course, not all mutations are recessive. You can have mutations that produce toxic proteins that interfere with some other aspect of the cell. And those are dominant, and those, that doesn't apply here, but the majority are recessive. So on average, and this is empirically the case, if you take two different populations carrying different sets of mutations and make a hybrid between them, you get heterosis, you get an increase in fitness. And that would have happened with the first haploid cells coming together to make it. So this is fine, but the problem is we've only explained one half of the life cycle. Sorry. Right. That's fine. Yes. Yes. Yeah. Yes. Yeah. So actually, a lot of the things that make organisms robust may not be robustness to mutation, but robustness to translation error or environmental insults and stuff like that. But the other point is that there are indeed all sorts of features that mask the effects of mutations, redundancy and chaperones and this and that. And there was talk yesterday about these sort of mechanisms. But it's still true, however fancy your cellular machinery, there are still going to be mutations of deleterious. And part of the problem, actually, in evolutionary terms, is that the fancier the cellular machinery, the more frequent these mutations become. So if you mask the effect of mutation, then it actually just increases in frequency. So it's sort of not entirely clear what the consequences will be. Yeah, yeah. And And I think there's a bias towards us saying this evolved for some evolutionary reason because it, reduces mutation rate or increases the chance of a beneficial mutation. But there are lots of straightforward physiological reasons, as Bruce says, for these sorts of things. Yeah. Anyway, we'll come across these sorts of issues later. So that's a whole other issue in organisms with, parental care or where the cost of a loss, say, early in pregnancy is reduced. Then again, these are also processes that to some extent mask the effects of mutations. If it's the case that, as actually is the case for humans, that many early defects are actually lost as miscarriages before anyone even notices, and then the number of offspring you have is independent of those, then that does in fact sort of masks some mutational effects. But those mutations are still being eliminated, of course. So it is still selection. It's just that it's selection that doesn't impose a reproductive cost on the population. So it's a little bit different, actually. It's not that the effect of the mutation is masked, because that individual does not actually survive. Okay, so we've got half the explanation, which is pretty good going, actually. But the trouble is, then you say, what's the next step? Well, the diploid cells, they accumulate stuff, and they'd be better off as tetraploids. And then the tetraploids, they octoploids, and it would be great. But the cell eventually buckles under its own, the weight of its own genome. So, you need to explain then why then separate back into haploids. Well, it depends. And so this is part of the argument between Fisher and Wright. So let's take a very simplistic view of it, which is to say that the number of functional copies determines directly and in proportion the amount of enzyme activity. So what you imagine is some kind of graph here of the number of copies, which is a surrogate for enzyme activity, against some measure of fitness, which might be mediated by the flux through a pathway or whatever, and you typically have some kind of saturating curve. That if you have huge amounts of this enzyme, then you don't get any benefit because you're limited by some other step of the pathway. And so the point is that if you have, this much typically, and then you go down to half that, it's not much different. And actually, the measurements, there are good measurements of the average effect on fitness of a recessive lethal in Drosophila. You can identify mutations in Drosophila in natural populations which are lethal as homozygotes. As heterozygotes, they have a 1 or 2% effect on fitness. So that actually is a measure of the typical dominance. Right? And if you go down to 0, then you're dead. So this is the sort of extreme example. So it's this curvature, really, that gives you the recessiveness. You have a separate argument for why it is that you produce this much instead of this much normally. And then there are various arguments about margins of safety. So maybe the environment fluctuates. Normally, this is an excess, but actually, you need to have some reserves or whatever. So it gets kind of complicated. When you go through detailed physiological models of biochemical networks, metabolic networks, then you do find that you get typically recessive effects. Okay, so how do we explain the separation of the diploid into two haploids? How do we explain the origin of something like meiosis? Because if they just separate without anything having happened, then you just uncover all the deleterious mutations again, and you're kind of stuck. So what's going on here? Well, basically, you can think of two kinds of advantage which might arise. One is an advantage from the possibility of repair. And the idea is it's very simple, that if you imagine a diploid organism, well, no, let's start with a haploid organism, start with the simplest case. And you have a DNA double helix, okay? So you have two strands. If you have a break or some other kind of defect in one strand, that can be repaired because there are a whole lot of enzymes which will recognize a mismatch or a single break in a DNA double helix. So that those repair mechanisms mostly work, okay? But if you have a haploid organism and a double-stranded break, well, you've lost the information in that region. If you've got a small deletion or you've just had a break in the chromosome, it's kind of stuck and it can't replicate the DNA. So that's fatal. But in a diploid, you've got 2 copies of everything. And you can now use information from the homologous strand to repair this double-stranded break. And it's astonishing that these repair mechanisms are so efficient. And one very striking line of argument is that if you take human tissue culture cells and you irradiate them with UV and immediately force them to condense their chromosomes artificially, you see there are lots and lots of double-strand breaks. Complete disaster. But if you irradiate them and just leave them be for an hour or 2, they're fine. Right? It's all repaired. So they're very efficient repair mechanisms for double-strand breaks, but they require diploiding. So the point is that diploid organisms are more robust to damage and can repair damage. So that's one advantage. And the second advantage is the one that I'll be focusing on, actually. And that is that if you have a diploid stage, then you have the possibility of recombination, of genetic exchange. And most familiar is the meiotic process, in which the 2 chromosomes pair up and they cross over at 1 or 2 points typically, so that you produce a chromosome that combines segments from the 2 parents. So that again produces novel combinations, and in particular, if you have a chromosome here with one deleterious mutation, another one with a deleterious mutation here, the crossover allows the possibility of recovering the intact copy, okay, by doing that. Of course, the crossover can also produce a much worse copy, but it produces variation, and selection can then at least pick up this intact copy. So recombination allows a kind of repair by selection. The double-strand break repair mechanisms allow direct repair using homologous information. So, is it recombination or repair or both? And actually, it's kind of both. Just to explain this, maybe it's worth, and I feel bad actually, because I haven't mentioned bacteria very much. I've been focusing on we eukaryotes. I should feel better about this, because the evolution textbook I wrote with Jonathan Eisen, who's a real bacterial phylogenetics guy, and he really stood up for the bacteria and the archaea, and insisted we had to have a chapter on the bacteria, a chapter on the archaea, a chapter on the eukaryotes. So it was, equal balanced text. Very rare to get one of these really balanced textbooks. Sorry? Yeah, but not all mutations are repaired. So sometimes there are mistakes. So if repair mechanisms were perfect, we wouldn't have evolution, actually. I mean, nothing would ever change. So this goes back to the whole evolution and mutation rate issue. So I'm not saying anything's perfect, but I'm saying that at least you have the possibility of evolving double-strand break repair if you have diploid. If you're haploid, you've simply lost the information. There's no way to repair it. And you can cobble things together and hope for the best, but it's not good. Yeah. Yes. Most eukaryotes, I should say. Indeed, that's a good point. And I sort of was hoping to avoid this somewhat difficult question. All I'm going to do here is talk about possible factors that may have been involved in the origin of the haploid-diploid cycle and of sexual reproduction. Okay? And one factor is that it's good to bring distinct haploids together because that masks the effects of deleterious recessives. A second counterbalancing factor is that haploid cells may be able to just reproduce faster than diploid cells. Okay? There are all sorts of life history peculiarities, which factors which may work in either direction. And there are models around which are consistent and make sense. I don't know if they're the real explanation or not, but one possibility is that actually what may evolve is an alternation between haploid and diploid life cycles. A brief period of diploidy allows repair of damage, and then a period of haploidy allows faster growth. So when I say repair of damage, either physical repair of double-stranded breaks or repair by producing recombinants, which can then be selected. Okay? Actually, it's quite an intricate and difficult, well, not difficult, but it's an interesting area, the haploid-diploid evolution. And one of the features of it is that actually what evolves is not at all necessarily what's optimal, what's best for the species. Probably shouldn't get into it in too much detail, but just to say that There are models which seem plausible and which could explain the origin of haploid, diploid, alternating phases. They're not necessarily optimality models. Sorry. Yeah, so exactly. We'll be coming to this in the later lectures. It's primarily if you have recombination, that allows the production of an intact copy, and therefore the selection of a better genotype in the end. But it's actually quite an intricate argument, which I'll be going through in more detail. But yes, essentially, sexual reproduction, recombination allow the more efficient purging of deleterious mutations. Yeah. Yep. Yeah, absolutely, absolutely. That's another argument. So again, I'll be going through this later. Sex, you could say, as Weissman said in 1900, a long time ago, that sex makes selection more efficient, both in getting rid of bad mutations and in establishing good mutations. And both of those can be substantial effects. And actually, we don't know the relative importance, but I'll be trying to explain that later. Yeah. What I'm doing at the moment, to try and sort of give some sort of logic to it, is to think about how the whole thing got started, and not to come to any conclusions because we don't know the conclusion, but also to raise this issue that actually recombination involves, evolution of sex, evolution of diploidy, may have involved repair as a selective process, as well as recombination. So to explain what I'm getting at, think about recombination in bacteria, because bacteria do have recombination, and it's really important. A lot of antibiotic resistance, is a problem because antibiotic resistance can be transferred from one bacterial type to very distantly related bacteria. So recombination is good for the bacteria and it's bad for us. But how does it happen? Well, actually, sort of three different ways. One is so-called transformation, which is simply the uptake of fragments of DNA from the outside environment, from dead bacteria or whatever, and their incorporation into the cell and very occasionally into the host genome. Second is transduction, in which a virus of a bacterium, and viruses of bacteria are called bacteriophage or just phage. So this virus will infect the bacterium and replicate and then produce offspring, lyse the bacterium, etc. It may actually incorporate into the host genome, and when it excises again, it may actually take a bit of the host genome with it. So just as part of the whole viral infection process, it can carry bits of host DNA from one place to another. And finally, conjugation, which is somewhat similar. Plasmids are kind of extra chromosomes that are found in many bacteria and which have an independent life cycle in a way and can transfer by conjugation from one bacterium to another. And they also can carry parts of the bacterial genome from one bacterium to another. Now, all of these seem to be actually kind of not necessarily adaptations for sex in the way that we see meiosis as something which has a definite function for sexual reproduction. These kind of processes in bacteria arguably are simply there for other reasons. So obviously with phage and plasmids, with transduction and conjugation, it's to do with passing on an infection, passing on a plasmid. Transformation people argue about a lot because there are specific channels for taking up DNA, but it's just food, right? There's a nutrient source out there. You take up DNA, it's good, you break it up, you use the base pairs, etc., bases, but by accident, bits of DNA are transferred. And the rates of transfer are actually very low, very, very low. It's just that bacterial populations are so big that the effect of a very rare recombination event can be very important. So the sort of simplest view of things would be that in bacteria and viruses, recombination is simply a side effect of the life cycle. And more generally, people have argued that maybe the whole origin of recombination was actually a side effect of repair processes. Because when you repair double-stranded breaks, right, you can get a recombination. So suppose we have, this is, I'm not very good at drawing this. This is one DNA helix with a double-stranded break, and this is another one, okay, which is intact. So normally, you simply have a repair of this using the information from here. But maybe you get the production of this, okay, in the process, which is a recombinant. So maybe the repair process doesn't quite work precisely, and you get a recombinant produced. In the same sort of way, you get these sort of accidental side effects in bacterial archaeal combination. Yeah. Yes. Well, if there's a double strand break, and if you've aligned the chromosomes, suppose you've lost 3 bases here, and there's a gap, then you do know, in principle, I mean, I don't know how the enzymes actually do it, because it's very clever, but there's a break, and there's an intact strand, so you simply copy from the intact strand. That makes sense. It's true. Yeah, then you don't know. So if you've got one strand which has a mutation which is perfectly well base paired, there's no way of knowing that it's bad or good. Right? That's actually a basic principle of evolution and genetics, that mutations are random with respect to adaptation, and the repair process is also random. They don't say, is it good to look up the genetic code and look up what this protein does, and go to the textbooks, and they say, oh, we don't want this here, right? There's no way to do that. But it is possible to say, well, deletions are bad. We don't like these. I mean, again, it's not obvious whether if you have a mutation that produces an insertion, so that now you've got, here's one genome. I'm not going to try and draw double helices again. And here you've got a little insertion. Now, you don't know which one is right, whether it was an insertion or a deletion. And there tend to be repair processes which repair it, but they're biased towards, let's say, deletion. Right? Now, maybe mostly that's the right thing to do, but, it's... Yeah. Yeah. Yes, one or the other at random. I think you do have to repair it, because otherwise you get this, well, anyway. No, I suppose that's right. If it's, yeah, you could just read through. So, yeah. Anyway. Yeah. Yeah. So, yeah, I'm not sure the answer to that, because actually... Yeah, yeah. So transposons are, as part of their life cycle, they have to insert. Another way to look at it is to say that actually mutation rates in some bacteria are, lower than in eukaryotes, and the mutation rate reflects the efficiency of repair in some sense. So, yeah, it's a complicated question. So I suspect that if you asked a eukaryote geneticist, they'd say the eukaryotes did it better, and a bacterial geneticist would defend the bacteria. Yeah, it's hard to. Anyway, the point I want to make is that the origin of sex, recombination, haploidy, diploidy, all of that may well have been very much tied up with repair processes. And indeed, the repair of double-stranded damage may involve as a side effect crossing over. So at the origin, it's very murky and unclear. But what I want to focus on is the current maintenance of meiotic recombination in eukaryotes, which is simply, it's not that it's a more interesting question, but it's a question we can actually tackle, because we can do experiments here and now in present-day populations. And firstly, just a kind of intuition, which I think is actually rather hard to defend, but it is that meiosis looks like an adaptation. The whole business of sexual reproduction looks as though it's there for sexual reproduction rather than a side effect of something else. But that's not a very kosher argument. A very strong argument, I think, is that actually the process of meiotic recombination involves enzymes which have their origin in the repair system, but have been now changed their function to actually make double-stranded breaks in order to allow a crossover. The process of crossover must actually require cutting both strands. And there are enzymes that come along and make double-stranded breaks at the homologous sites and allow crossover. Okay? Now that doesn't look like repair. That's creating damage and then using the damage, repairing it to make a crossover. And also, many organisms lack recombination. Okay, male Drosophila, most famously. One of the reasons Drosophila is very good as a genetic organism is that in males, the chromosomes pair up, but they actually don't cross over. They have some fancy mechanism of their own that allows them to segregate without crossing over. Now, if recombination was somehow necessary for repair, then male Drosophila will have an enormous mutation rate, and they don't. So certainly recombination isn't necessary for repair, and it seems as though the whole business of eukaryotic meiotic cells seems to be an adaptation in which double-stranded breaks are actually made in order to produce recombinant chromosomes. So what I'm going to focus on for the rest of the time, how am I doing, 24 minutes? Okay. So what I'm going to focus on for the rest of the time is explaining eukaryotic regular sex, which is not to say all the rest of it isn't interesting, not to say that the origin of the whole business isn't interesting, but it's just less attractive. Yeah. Mm-hmm. You mean about the origin or about the present day? Mm. Well, they sort of argue, but it's not completely irrational argument. So, I mean, it's not really my area. I like, solid population genetic models and things you can test in present-day population, this kind of thing. But actually, people do make rational arguments about even the origin of life, or even things like why we have 4 bases in the genetic alphabet and not 2 or not 6. And actually, the hypotheses are not, testable experimentally, but there are observations you can make that suggest that one or other is more or less plausible. Comparative studies, but also when you're looking at sort of origin of life questions, origin of the genetic alphabet, it doesn't do any good to compare things because this is stuff that's common to all living organisms. Right? Maybe I could take an example from this paper by Satmari on the genetic alphabet. Kind of clever. So what he does is just say, well, one possible explanation is that there's a trade-off between having a low copy error rate. Clearly, if you just have to align A with T, the rate of error is going to be lower. It's an easier task than if you've got 8 possible bases and you've got to, pair the right ones up. That's going to be harder. Versus in the RNA world, a world before DNA, before the whole genetic code evolved, when RNA molecules were doing the whole business. They were replicating and they were also carrying out catalytic activities. Now, if you've only got A or T, you can't make a very interesting RNA molecule. It can't really do much. It turns out, amazingly, that if you have A, T, G, or C, or I should say A, U, G, or C for RNA, if you have those 4 possibilities, then you can produce chemically very sophisticated molecules that can fold in different ways and catalyze reactions, including their own replication. So the RNA world could function with 4, but you could say, why not have 8? Then you could have even more. And you could say that an argument for the evolution of the whole genetic code and the whole move to proteins doing the work is proteins have 20 chemically very different side chains, amino acids, and they can do far more catalytically. So if you want to make a for the good of the species argument, then we're better off with our protein-based system because it's chemically flexible because of the separation of replication, 4 bases, but from catalytic activity, et cetera, in the proteins. Okay, so there's this trade-off, and actually Satmari sort of makes some quantitative arguments based not just on sort of verbal reasoning, but also on experiments. People can do experiments producing non-standard bases and producing genetic alphabets with 6 or 8 letters and measuring the error rates and looking at the chemical flexibility and so on. That was a bit rough and ready, but you can make a rational argument there that 4 is a pretty good number. Whether that counts as really testing hypothesis, I don't know. It's not just hot air. Okay, so I go back. I'm actually sort of going to slightly row back from my really individual selection-based arguments to say that actually there is selection amongst species for sexual reproduction. And so the early arguments that suppose that sex evolved for the good of the species aren't completely wrong. And just to illustrate this, to make the general point that actually sex has very often been lost. It's not at all uncommon for organisms to go from outcrossing sexual reproduction either to selfing, very common in plants, for example, or to some kind of completely asexual reproduction. And they can do very well in the short term. In the long term, those species seem to die out and not to leave progenitor species, with a very few, very rare and very puzzling exceptions. So Maynard Smith made a very nice example, which involves these rather horrible terms here. It's actually a very neat comparison. There are two kinds of parthenogenesis involving the development of an individual from an unfertilized egg, and this will be a haploid individual. Well, unless there's a doubling up to produce a diploid, which can happen as well. But we say that, I don't know where these terms came from, but orinatoky is when a male develops from an unfertilized egg. And this is actually typical in bees and ants and so on, and in many, many other groups. It's actually evolved in the insects about 8 times, and 5 of those at least have produced really big groups, like the bees or the ants. And what happens in these so-called haplodiploids is that the females are diploid and the males are haploid. And then they can mate. The females have meiosis perfectly normally. The males simply produce gametes directly. If you work through it, you realize that this is simply like having one big sex chromosome. So that we have, males have 1 X chromosome, females have 2 X chromosomes. In haplodiploids, because the males develop from the unfertilized egg, essentially the whole genome is haploid, right, in the males. And this actually has an important consequence, which means that the female can control the sex ratio. And a lot of really interesting work, arguably some of the most precise quantitative work in evolutionary biology, has been based on sex ratios in Hymenoptera, where the female actually adjusts the sex ratio, the ratio of males to females, to an evolutionary optimum. And they're very, very elegant predictions developed by Bill Hamilton originally and confirmed by him, mostly working with big wasps, which have this haplodiploid system, showing that the sex ratio really does adjust to what the theory says it should. So this is very neat stuff. But, of course, it's not parthenogenesis. I mean, you produce males by asexual reproduction, essentially. The female just produces male offspring with no fertilization. But, it's a male. Right? What do you do? And actually, turkey breeders tried to produce asexually reproducing turkeys. This was way back in the 60s. And they managed to get some, but unfortunately, and they should have realized this before, because the way sex chromosomes work in birds, they're all male offspring, so that was a bit of a waste of time. But philitoky is the opposite, and you imagine mechanistically there's no particular reason why you shouldn't be able to produce, leaving aside sex chromosomes. Sorry. No, the female produces eggs, which are haploid. If those eggs are not fertilized, they develop as males. If they're fertilized by a male, no, I'm thinking of the X chromosome. So these don't have sex chromosomes, right? These simply have the ordinary, autosomes, right? So they have the females have 2 copies of everything. They can go through meiosis, producing eggs that are haploid, have 1 copy of everything. If those eggs are not fertilized, they develop to males. So the males are all haploid. If they are fertilized, then they have the male genome coming in, and they're diploid, and they develop as diploid females. Okay. So that's one way of doing this. But you could have the opposite, which is where the unfertilized eggs develop as females, and then you can have continual asexual reproduction, right? You just have female, 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 female. And this has arisen many, many times. It's called philitoky. But it's somehow very sporadically distributed. It's never produced a big family, a successful family of species. Okay, it's just produced species that may persist for maybe hundreds of thousands or a million years, then go extinct. Okay, so this is a really striking contrast, and it seems to indicate, and it's a very, very common pattern, that asexual reproduction may work in the short term, but in the long term, meaning million-year timescales, it's a disaster. It's not going to go. And this is just a sort of illustration of the kind of phylogenetic pattern that you typically see. You do see asexuals, but they're at the tips of the tree, right? Maybe they diversify to 1 or 2 species, but they're just at the tips. They don't have a long-term future. And these are just some examples of very successful asexuals. This is Nematophorus, which is an all-female lizard in South America. These are Pachyliopsis, top minnows, I think, which live in Central America, which are interesting. There's an all-female species which still has to be fertilized by males from another species, but the sperm are discarded. Okay, so it's a cheat. It's unfair. Okay. This is yeah. Sorry. Yeah. Where's this question? Yeah. Okay, so I shouldn't really call it asexual, but everyone else calls it asexual. So you're right that it depends how it's produced, actually, and there are different forms. So I think, actually, in the examples I gave, it really is asexual because the offspring are genetically identical to the mother. It can go both ways. You can either have what's equivalent to selfing, I can't remember the technical term now, in which heterozygotes produce half heterozygotes and half homozygotes. That can happen. Or you can have a system where the offspring are genetically identical to the mother. It just depends how the detailed parthenogenesis works. So actually, the long-term consequences evolutionarily are pretty much the same because very quickly the selfers become genetically identical. So I haven't been making the distinction, but you're right, I did make the distinction earlier on. And you're right that strictly speaking, if it's a mechanism which involves meiosis still, then it's still sexual, but it's not outcrossing. So, can't remember. I should remember these because I did a whole lot of work. This is an alpine plant whose name I've forgotten, which at high altitudes, where there are a few pollinators, will actually reproduce parthenogenetically. Okay? That's not by selfing, it's actually by producing genetically identical offspring. I'm having a blank out. Can anyone remember something? Yeah, Bruce, you're a natural history guy. These are these Darwinula, I think they're called. No, anyway, I will not describe this one. I'll describe the rotifers, which are one exception. There are many groups of these rotifers, which are very multicellular, but very small animals, pond animals, which appear to be, one group of those so-called diploid rotifers appears to be asexual and appears to have been asexual for tens and tens of millions of years. There's no record of males being produced. And this is actually still very contentious and very obscure, but it does actually seem to be a long-term asexual. People still are arguing about how it's avoided the apparently inevitable deleterious consequences of sex. But that's very much an exception. These other species are very recently originated. It's actually, actually some of them have quite a lot of variation because some of these are produced by what's called hybridogenesis. So when you get hybridization between two sexual species, sometimes there's quite a high frequency of producing parthenogenetic hybrid offspring. And those offspring have the advantage of heterosis, right? Because they're bringing together two genomes. And they preserve that advantage by reproducing asexually. So they do very well in the short term. In the long term, things go downhill, but we're talking about things that are relatively recently originated. And some of these originated in multiple origins, so there's actually quite a lot of genetic diversity as well. But it's still clear that in the long run, that diversity will diminish. It's a very good question. And so there are arguments that diploid rotifers particularly seem to be able to avoid sex because they have amazingly efficient repair mechanisms. And they have a life cycle which involves surviving complete desiccation. Right? So they're incredibly radiation resistant, which is a side effect of being desiccation resistant. You can mash up their chromosomes, they'll repair them, And so it seems that these repair mechanisms may have reduced the mutation rate. I don't think there's a mutation rate measure. It's actually quite hard to do. And these recent asexuals are probably too recent for things to have changed very much. So, yeah. Yeah. So there's a very interesting pattern, which is actually, I don't think there's an accepted explanation of it, which is that asexuals often originate and spread at the edge of the range. So, a species will have a range out to here, and then the asexuals will live outside that range. So here, I think it's Antinaria. It's plant lives at high altitudes. And there you can see why it's at the edge of the range, because asexuality is a big advantage if there are just no pollinators around. What else can you do? Another reason I'll come to later, when there actually is a very strong and immediate reproductive advantage to being asexual, which is that a female produces 2 female offspring. So the reproductive rate of the population is actually twice as great, because you don't waste time producing males who are kind of useless. So yes, there are interesting patterns to the prevalence of sex. Okay, so what I want to finish by doing is to say that actually there has to be a short-term advantage. It's no good saying that, well, in the long run, it was a mistake going asexual, because, you know, everything will go asexual. Asexual variants would arise, and they don't seem to be major obstacles to their origin, at least in some groups. And they would simply displace everything. And even if in the long term it was a mistake, well, you know, it doesn't explain why sexual reproduction is persisting and is maintained at high frequency. A number of slightly more detailed arguments there. One is that you have many groups that have cyclical parthenogenesis, aphids, gall wasps, and so on. Aphids, for example, will go through asexual reproduction for 5 to 10 generations over the summer, and then in the winter they'll go through a different stage, a resting stage, which is produced sexually. So sexuality is an essential part of the whole life cycle through the year, but there are a number of asexual generations interspersed with sexual. Same for Daphnia, which is a small crustacean living in ponds. Many, many cyclical parthenogens. So although I say eukaryotes have obligate sexual reproduction, that's only at some stage, not necessarily every generation. There's also facultative parthenogenesis, meaning simply that, you know, there can be sexual reproduction only under certain environmental conditions, often associated with harsher conditions or with dispersal. Species of nematodes or snail Potamopergus, which is in New Zealand, which under certain conditions will reproduce sexually, but mostly reproduces asexually. And another kind of argument, not about sexual reproduction, but about recombination, is that actually there's genetic variability in recombination rate. You can take Drosophila populations and select for higher recombination and select for lower recombination. So you can ask, you know, why is it where it is? Why is it at the rather high rate that we see? Because if, you know, all the reasons I'll come to, recombination is actually costly, you know, why maintain it? So rates of sex, rates of recombination are at certain rather high levels. Why? We can ask that question about current populations. Now, the advantage of sex and recombination really have to be quite strong. I mean, it's kind of obvious that it's complicated. Meiosis is complicated. It's much slower, much more costly energetically. You know, why not just go through the much simpler mitotic process? At the level of whole organisms, multicellular organisms, courtship and mating are risky. Actually, this applies to unicellular as well. It's a lot of trouble finding another individual. It limits your ability to colonize new habitats, etc. The most important reason, at least the most quantifiable reason, is the so-called twofold cost of sex, which applies if you have so-called anisogamy, if you have the traditional role of the female producing a large egg full of resources, the male producing a small motile gamete. And we can think of this contrasting role of the sexes either at the single cell level, a single-celled organism, there may still be a differentiation of large and small gametes, but at the level of the multicellular organism, if you're a female and produce a large egg, you typically also look after the young. This isn't always true. Anyone think of an example? A male seahorses. Yes, the seahorses are justly famous because the male, it's amazing that they mate sexually, but the female transfers the egg into the pouch, the roux pouch of the seahorse, who then gets pregnant and produces the offspring. So it's simply a reversal of roles. But normally, most of the time, the female devotes most resources to rearing the offspring. Now, in that situation, basically a single female in a population that is stable can produce 2 daughters, whereas in a sexual population that's stable, in which the male mates with the female, it's the female producing the resources, so she produces 2 offspring, of which one is female and one is male. So with anisogamy, basically a variant that reproduces asexually and produces a genetically identical female offspring will increase at a factor of 2 from low frequency. And this is the twofold cost. It's important to realize that it depends on this asymmetry between the sexes. But given that asymmetry, which is very common, we expect these asexuals to increase twice the rate. And in fact, that may be the explanation for why some of those asexual examples I gave can expand their range beyond the limits of reproductive ability of the sexual species. There are other costs to recombination, for example. If you think about what selection's doing, it's building up good gene combinations, and what recombination does is shuffle them up. And in the short term, you would imagine that would always reduce fitness. And in fact, one can show mathematically that if you have a constant fitness of every possible gene combination, then because selection builds up the fittest gene combinations, and because recombination has an essentially random effect, it will always reduce, on average, the mean fitness of the population. This is the so-called reduction principle. And one can actually measure that. It's rather hard to do. There have only been a few rather heroic experiments in Drosophila, one by the Charlesworths, one by Loci, I think, quite a long time ago. But they basically use the trickery of Drosophila classical genetic engineering, not just ingenious crosses, basically, to produce, basically take chromosomes from the wild and either pass them through males, in which case there's no recombination, and look at the effect of that chromosome on the survival of larvae, or take a chromosome from the wild, take it from a female, therefore it would have undergone one round of recombination, and ask for the effect of that recombined chromosome. So you do a side-by-side comparison, the effect of a chromosome sampled from the wild with or without crossing over, and ask about the average viability of the offspring. And there's a kind of a 2% greater viability if the chromosome hasn't gone through crossing over. So there's a small but measurable recombination load. And this has been repeated a few times, but it's actually a lot of work. It's not a large effect, but it is an effect. Well, yeah, so a lot of the arguments for why sexual recombination might be favored is that, okay, in that environment, it was good to keep to the old gene combination. In a new environment, it may be good to change to a new combination. And that's a lot of the argument about why sex might be favored. So this is only a cost in a stable environment. That's a good point. Finally, we can see examples where clearly selection has acted to reduce recombination. And one of those is in sex chromosome evolution. This is the clearest example. So sex chromosomes can originate very easily. So suppose you imagine an organism that has, let's say, it's common many insects, a single X in the male, and in the female, 2 X chromosomes. So this is the X chromosome, these are the autosomes. Now, suppose there's a fusion between the X chromosome and one of the other chromosomes. This can happen rather easily. So now what you have is something that is going to act just like a Y chromosome, and you've got a new X chromosome that's twice as big. Okay. And what happens is that there's very strong selection for alleles on this new Y chromosome to be favored in males. This Y chromosome now this chromosome suddenly finds itself only ever in males. And so there's strong selection for the alleles carried on this chromosome to be fit in males. And for the genes carried on this chromosome, originally these were actually, homologous. Genes on this are under selection to be good in females as well as males, because these are found, some of the time in females, some of the time in males. So this leads to very strong linkage disequilibrium between these genes, sort of male-favored genes here, female-favored genes here to some extent, and crossing over here will bring genes that have been adapted for one sex onto a chromosome that's found in the other sex. So what happens in sex chromosome evolution is that initially you just have a fusion of 2 chromosomes producing a new sex chromosome. Initially there is recombination, but that recombination is deleterious because it basically breaks up combinations of genes that are favored in one sex rather than the other sex. So there's very strong selection for suppression of recombination. Yes. So, well, we actually know the timescale because in Drosophila there's been recent work by Doris Batrog and others which actually trace the degeneration of sex chromosomes and the reduction in recombination that's occurred. You can actually see this in mammalian sex chromosomes as well and bird sex chromosomes. But there seem to be a series of events in which recombination is reduced and which gene function is lost. And you can get at this by looking at comparisons across species. And the timescale is of the order of millions of years. So this doesn't happen overnight. But over a million-year timescale, you get reduction in recombination, and as a consequence of that, for reasons I'm going to explain later in the lectures, you get loss of gene function. And eventually, the Y chromosome may disappear altogether. Yes, yes, that's right, yes. So initially, they're just ordinary chromosomes. All that's happened is a fusion, that's it. But now you've got a different selective environment, so selection pressures are acting in different directions on the two chromosomes. This leads to selection to reduce recombination. Once you reduce recombination, then you become essentially a bit of asexual genome. And then for reasons I'm going to explain later, this is kind of a bad thing. And for the same reasons that asexual species go into decline, sex chromosomes go into decline. And you can actually measure the rate at which this happens. And there are similar examples, a bit less dramatic, but in sticklebacks, there have been multiple adaptations of this marine species to freshwater lakes after the glaciers retreated. So this is over a few thousand years. And there's been really beautiful work looking at the genetic basis of these adaptations. And it seems that many of these adaptations involve sets of genes which are linked together. And that may be, it's likely to be, because if you have a set of alleles that are adapted to freshwater, and if there's still some hybridization between the marine and freshwater in estuaries or lake outlets or whatever, then you'll have selection to reduce recombination between the sets of genes good in this environment and the sets of genes good in that environment. So it's another example, rather like the 6 chromosome example, where selection favors reduced recombination to keep intact combinations of genes favored in different environments. And another example, which I think people here are working. I fell off the end of the presentation. Anyway, the last example I was going to give was the so-called supergenes in papilio butterflies, which have multiple mimetic forms, multiple wing patterns maintained to mimic different distatal model species. And these involve multiple genetic factors controlling different elements of pattern and behavior and whatever. And these are linked together very, very tightly. So again, selection has acted to produce a tightly linked cluster of genes. All of these are examples where recombination seems to be a bad thing, seems to have been eliminated by selection. So why do most organisms actually still recombine? And that's what will be explained later in the week. That's it. Yes. It's an interesting question. So actually lots of wild plant species are also polyploidy. So polyploidy seems to be rather easy. And they may be established by, again, the immediate advantage of heterosis. Most of these polyploid species, not all of them, but many of them, most of them, are produced by hybridization from different species. So there's an immediate advantage. There may also be physiological advantages. There seem to be immediate effects of having bigger cell size. So actually, I don't think people know exactly why polyploidy is so successful. It is. At lunchtime.